one to Dr. Jen McCormick, who is interviewing for our research ethics position, ethicist position here, and has graciously agreed to engage in this mock consult that I'll explain briefly. The purpose of this role, in part, is to develop a broader initiative for research ethics here at the institution, which includes a research ethics consultation service. In that regard, we have contrived, and I do mean contrived, ethics consultation that obviously in real life would be very different. Dr. McCormick would have more knowledge about the researcher and the protocol and so forth. But the purpose of this exercise is to see how candidates operationalize the extensive knowledge they have about research ethics when engaging with a faculty member who, while hopefully not typical for our institution, is the kind of person you might encounter in the capacity of a research ethicist. So I'm going to just read, if anyone wants a copy, we have some here, but I'll read the setup, then give Dr. McCormick an opportunity to ask any clarificatory questions about the setup, about the dynamics, and then we'll let the two of them interact. So, an investigator at the medical center has discovered a new gene that he believes is strongly associated with childhood cancer. He submits a protocol to the IRB to use de-identified state newborn screening blood spots to obtain DNA samples from all infants born in the state for the next 5 years. The goals of the study are several-fold. 1, to conduct genetic testing of all newborns to look for inherited disposition to childhood cancer. 2, to test a new sequencing methodology that is faster and cheaper than prior methods. And 3, to develop a biobank available to other researchers. He believes that the study should be exempt from IRB review and that consent is not necessary. The IRB has indicated that this protocol raises some serious ethical issues, and they suggest that the investigators seek research ethics consultation before moving forward. Reluctantly, he agrees. This is the first meeting between the ethics consultant with the investigator, whose main aim is simply to move forward as quickly as possible, as he has funding that begins in 4 weeks and a graduate student's clock is ticking. While aware of some of the ethical issues, he is not particularly concerned about them, noting that it is impossible to anticipate every eventuality and that a similar study was approved at his prior institution. The investigator is frustrated at this extra level of review and does not understand its function.
Do you have questions?
About sort of the setup.
Not about the setup.
Let's just, the one thing is that if we can, I don't know how much research you've done into it, let's hold in abeyance this rapidly changing field that suggests that prospective studies of this type will not be allowed in the future. So we really want to have a conversation about convincing me to do the right thing.
So, meaning don't consider the draft changes to the...
Oh, you can talk about them, but right now, based on some March of Dimes stuff that Sheila found, you could just say, you're not allowed to do that, and then the conversation's over. That just came on board. So I'd like to have, at least in this contrived story, enough wiggle room that you can convince me to do the right thing.
Okay.
Is that good by you?
Yep.
Warn her about the grumpiness, too.
I did. So I'm going to channel my inner curmudgeon. Try not to get physical, that's illegal, but don't take it personally. It's all good. We're friends, and even though we have an audience of thousands, it's going to be good. It's great fun.
Okay. So I like to begin a consult by, I guess I do have some questions about the protocol. And it sounds like, based on the note that you sent me, or that was sent to me, you're planning to use DNA obtained from newborn screening blood spots. So are you taking additional samples so when newborn screening is done, usually there's, in some cases there's a number of samples taken. So are you wanting to collect an additional sample for research on top of what will be used in the screening process, or just taking leftover from the screening process, or planning to try to extract the DNA from the dried blood spot?
Is it okay if I just back up a little and tell you the story? So my student and I have been doing some studies studying childhood leukemia, and what we find is that there's a unique upregulation, or the upregulation of a unique gene that's in the FOS and Jun family of transcription factors that we call JOS. And it appears to be uniquely associated, this upregulation with childhood leukemias. That's all part of our ongoing IRB approved study. And what I'd like to do is, I'd like to map that gene looking at heel stick samples coming from kids that are registered with the state. The idea being that as I screen over here, students who have childhood leukemia, I can map variants, genetic variants, to the population. And I certainly can do this using the 1,000 Genomes Project, which we'll do anyway, but also I'd like this unique South Central PA population to kind of get an allelic frequency for variants and then map what we see clinically. So to your point, this is, I've talked to a casual acquaintance over at the state, and I can get their samples, no extra samples, totally de-identified. I have no interest in knowing the names nor following these people moving forward. I would just like to create a unique database in which we genotype this JOS gene, as well as using this targeted exome sequencing that we've developed, 150 or so of the usual suspects, the BRCAs and the MICs and those kinds of things, and then establish a database from that to which we can map the variants in this gene that we've discovered. So I'm really, I won't say upset about it, but it makes no sense to me. This is just de-identified. I have no interest in knowing the identities nor mapping back to them. And so I don't understand why the IRB is pushing me on this. It just seems really straightforward.
So, but you are wanting to, so I want to kind of go back to the technology, and the reason I'm asking this because when I read the note, one of the first things that came to mind to me was the issue of return of results. And this might be one of the issues that is getting the IRB nervous.
Well, the only way I could return any results I get is if I happen to know who the individual was, and I actually have no interest in that.
But it sounds, which, there is a lot of debate in the research genetics literature on return of results in terms of what should be returned, should anything be returned, what's the responsibility of the researcher. But it does sound like you are actually, when you actually said the usual suspects, so you will be looking, actually actively sequencing, looking for genes that are pretty well known and established to be associated with cancer and increased risk of cancer.
Right.
Correct. Okay.
Right.
All
But
right.
again, in a de-identified fashion. So, I mean, is there some responsibility now that I have to reach out and try and map a mutation with an individual?
Well, so that is something that there's a number of recommendations that have been put out in the context of genetic, and I'm kind of differentiating genetic research using this in this genetic scenario versus a clinical scenario, where no one comes out and says that there is a real strong responsibility, but there are some who would say that for things where it's a definite does increase the risk, and it's pretty well known, in other words, the penetrance is pretty high, that there is some desire on the part of participants to actually get that information, and the question as to whether you as a researcher should whether you actually don't have an obligation to return that. So, and
listen, we can do any kinds of what ifs you want, but the fact of the matter is anybody who does sequencing is at risk for having done that. Let's say that they're using deep sequencing technology to look for the CF gene, and they happen to see, if they were looking to see something over in a BRCA mutation. There is nothing incumbent upon them to go look at BRCA if they're in there looking at...
Right, so that would be an incidental finding. So, right, and it sounds like to me, though, that you're actually actively seeking and looking for those things. And most standard, what is becoming more standard in the research domain is if you're anticipating some of these findings coming up, that you think about a return of results plan. It sounds like you have absolutely no desire to get involved with that. And I think I would ask you to stop and really think about that, because if you are, if you do find some genes like BRCA, that's not, it has implications not just for the newborn, but for actually the parents, correct?
Right, well, that's a can of worms that I'm really not interested in getting into, and it's, the entire concern is abrogated by the fact that I will not know who these individuals are. I cannot follow them back.
So
What's the need to do that?
So I think the other issue that you have to realize is that this notion that genomic data can be de-identified or anonymized is actually not something that we can hang our hat on. There are ways to go back and...
Oh, absolutely. If I go and get the sequence of each of those 5,000 individuals, I could match them one to the other. There's no question about that. But this is another one of those what ifs. I have no inclination to do that. And what's starting to rankle me is the notion that I'm being accused of doing something. Really, all I want to do is advance our understanding of this childhood leukemia risk, not to try and provide any clinically actionable items. I'm not a CLIA certified lab. I can't do that.
So I don't know that you would necessarily have to have the results CLIA certified before returning them. There are, there are IRBs that have policies that do allow for the return of research findings without the CLIA certification, with clear clarification to the participant that, you know, if you want to follow up on this clinically, you will need to have it CLIA certified in a clinical setting at your own expense.
So you people are going to have to give me some guidance here, because what you've just described to me says that I'll need to consent to all 5,000 or so patients that I want to get. So you're going to have to give me some guidance.
I kind of want to step back a little bit on that as well. It's not clear to me that you actually can just use these samples for research purposes. A little digging around that I did do on this case, it sounds like some states don't really have any statements on whether they allow the newborns, the leftover spots, to be used for research or not. And the digging around that I did come across in the context of the state of Pennsylvania, it sounds like the newborn screening spots are not allowed for use for research.
So that's certainly not what I'm getting from my acquaintance in Harrisburg, but I could go to North Carolina and my friends at Wake Forest University and get access there. So the point is, I think I can find access to I'd prefer South Central PA, obviously, but right now I'm just anxious to get this project started and start building a database. So is there a path to make this happen?
So again, that's an interesting question. I would say, thinking about it from a socially responsible perspective, the path to do this is to really think about that you really should actually think about getting consent, really consider the fact that you are going to come up with findings, genetic findings, that may have real clinical importance to the individuals, their families from which those are obtained, and whether there isn't some type of ethical obligation or just your own sense that you will have knowledge about individuals that may be important for them clinically.
Yeah, but there are tons of folks that really aren't interested in knowing that, and so.
But I think the other thing is you typically don't have to hand that information to the individual. It's really merely a matter of offering.
Yeah.
So, for example, at an institution that I have been at, we have a biobank, and it sounds like you're just creating a database, not of the actual samples, but of the DNA sequence, correct? So you're not actually collecting the samples.
Well, no, what I'd like to do is maintain the libraries that we construct for each individual, so that if other individuals want to come and map their gene onto polymorphisms that we observe in jobs, they'll be able to do the sequencing for their gene.
But it's the DNA, it's not
Correct.
the blood specimen.
Correct.
Okay.
Correct.
So in this context, it is an actual biospecimen biobank. And when individuals are consented to, they do provide consent to participate in the study, and what they're providing is a very broad consent. So they're agreeing to the fact that their sample will be used for research that is unspecified, that it will be continued to be used until the sample runs out, that the sample or the data will be shared broadly. It'll be de-identified, but if you read most consents, you'll see that there's always a caveat, even though you're kind of couching it as a what if, the caveat that there is a possibility for samples or the DNA to be re-identified. And I think we have to come to terms with the fact that that is a little bit more of a reality than we like to appreciate, just with the increasing numbers of databases that are popping up. Right now, many databases are in silos, but eventually those databases are going to be cross-talking. And so there is the possibility of re-identification.
Anything is possible, but we're creating these obstructionist things that are going to keep us from moving the field forward. All of these initiatives, for instance, Vanderbilt, my understanding is, they have a program where it's just, you need to opt out. We're going to collect your samples, they're de-identified, and we're not going to map them back later. And so why can't we have that kind of a situation?
I think the other thing we have to realize is that they're given the opportunity, well, in the newborn screening, they're also given the opportunity to opt out. So that is analogous. I know enough about the Vanderbilt program to know that it's gotten a lot of flack over the years for the opt-out, its opt-out approach. Studies, empirical data show, some of my own research suggests that individuals are really pretty comfortable having their samples used in research. They just want to know. And so
well, that's certainly the case when I approach these families with childhood leukemia. I don't think I've ever had someone deny us the opportunity.
Right, so I think what you need to think, I would ask you to think about, is the fact that we need to be, in the biomedical sciences, trust is very important. And part of that means transparency. And this idea of using samples that were originally collected for clinical purposes, for one particular newborn screening purposes, one particular clinical purpose, and in which, yes, parents are able to opt out, but they're not even really, in most newborn screening programs, I think a lot of parents aren't even aware of the fact that the newborn screening is happening until it's done. They aren't really aware of the fact that the samples are being stored, and for how long those might be stored. And I don't know if you're familiar with some of the cases that have happened in a couple of the states, Minnesota being one, Texas being another, where privacy advocates really came down hard, and that caused state legislature to change some of the how and what newborn screening samples could be used for. It has really kind of limited the use of those samples in those states. So, right now, based on what you're telling me and based on the little bit that I know about the Pennsylvania law and the use and storage for newborn screening samples, and how the IRB at this institution typically deals with genetic and genomic studies, biobank studies, I would suggest that you maybe rethink, step back, and think about how you might be able to go about at least obtaining some type of consent for the use of the samples in research.
Yeah, well, we're just a small molecular biology lab. My budget's not going to support a nurse coordinator to go and consent this. My student can't do this. There's got to be another pathway to just generate the database.
You can actually ask for a waiver of consent in terms of when obtaining consent is not possible. And I think that may be applicable here. I still think that the IRB could potentially, what might be making them nervous is this idea of the return of results and the fact that you will be generating a huge data pool of information that could be significant, have some significant clinical impact for individuals.
Agreed. If I know who the individuals are, you're absolutely right.
But I think, it's not, if you're getting de-identified samples, you may not have the link, but you will be able to go back to the state. And I've actually been involved in cases where an individual at UCSF had a database, had just de-identified samples and had done some bioinformatics, genomics type analysis on the DNA, had no plan for return of research results, and then lo and behold, in some of her analyses, identified a handful of participants who actually had, I think it was, she identified the gene for long QT syndrome. Came up as an incidental finding, was completely not expected. And she was the more she thought about it, the more uncomfortable she was sitting on that information, knowing the potential clinical implications for the individuals from which those samples came. And so, again, she hadn't, the samples were de-identified, the data were de-identified, so she hadn't obtained consent, but she was able to reach out to the colleague, and I believe the colleagues at Stanford, who had the linkers, and they worked through a process by which it didn't. I'm a big proponent of not forcing results on an individual.
Well, I can well imagine that email.
Yeah.
Hey, guess what we found?
Exactly.
And so that's why I'm not even interested in the honest broker approach.
Well, and so I don't think you need to approach it necessarily as, hey, look what we found. It's more of sending a letter, a very kind of vanilla, plain vanilla letter talking about how your sample was used in a particular way, something has been found that may be of interest to you or your family members. If you would like to learn more, please contact us at such and such and such. Now that does obviously can cause some alarm to some individuals. I mean, when they're automatically, when they're getting a letter just kind of out of the blue. And I think I'd have to think more thoroughly about this, but it's this fact that you have samples that you're using for research for which individuals aren't aware of the fact that you are using them for research. I really don't, even though samples would be, or the DNA would be de-identified, technically that should not be considered to fall under the purview of the IRB in terms of being human subjects. The way we define a human participant is right now there's an identifier. I still think kind of going back, looking beyond the regulations and thinking about this issue of transparency, social responsibility. I mean, you could really kind of think of some unique ways, not you necessarily, but you in collaboration with your collaborator at the state to have even just information handouts, because you're collecting these samples prospectively. So just a sheet, an oral consent, kind of almost like an oral consent, a sheet saying your sample is going to be used in research purposes, for a particular research study. If you wish not to have this done, please notify.
Well, if that would satisfy the IRB, I think you're right. I think that's a fairly straightforward path. We'd have to look through that. Listen, I'm on the clock here. I've got to run, but Would it be okay with you if I sent my student by to talk to you about this language and we can explore this in more detail?
I have examples that we can use, and I actually, kind of thinking about this, talking more to you, I think the juices are flowing in my mind, and I actually kind of like this idea of an information sheet to the parents from whose samples you're taking, and then that would kind of alert them to the fact that should something be found in the future, that you oftentimes as experts, a group of experts deemed to be worthy of returning offering to participants, that they may be contacted, and at that point they'd have an opportunity to learn or not learn.
All right, well, that sounds like a plan. Listen, I've got to run. Jen, thank you so very much for your time.
Thank you.
Appreciate it.
So, obviously very good time. In your interaction with this also sensitive individual.
Thank you for that.
Yeah, you bet. What were your thoughts? What were you trying to accomplish in the limited period of time that you had with it?
I think what I wanted to know was to really get a better sense of what the goals of the study were, and then what the parameters were. And I also, I think, thinking, pausing, I think I felt a little slightly unprepared, because I don't know typically how the IRB here would handle these types of things. But one of the things was wanting to make him realize that my goal was not to hinder the research, but to really allow it to proceed, but to proceed in a way that engenders trust with potential participants and the public at large. And while researchers like to think that what they do stays in the laboratory and that their work is kind of isolated from social views, social policies, social perspectives, that's false. And I think what he was proposing, you can see a New York Times headline: investigator at Penn State is using samples collected from newborns to do research without having obtained consent. That's not something an institution would like to see on their website. And whether that's a little too pragmatic in terms of thinking, oftentimes being pragmatic is what can get investigators to realize that it is kind of important to step back and think about some of the broader social, ethical issues.
And as you think about what you would do differently if this was, you were here in this position, how might you prepare or approach this encounter knowing it was coming differently?
So I would want to talk to the folks at the IRB who had actually referred him to me to get a sense of what their concerns were, and make certain that I think it would be very helpful to get a sense of what their concerns are. One of the things that's really frustrating for investigators, myself included, when I have to submit IRB protocols, is when one person is saying one thing, you're going to one person for advice and they say one thing, you go to another person for advice and they say another thing, and you go to yet a third person for seeking advice to try to resolve the two different pieces, and you get yet a third piece. And so I think I may not be on the same page with the IRB, but actually trying to get a sense of what their concerns were before going to Dr. Vrena and saying one thing that completely contradicts and would be out of left field for the IRB. So that's one thing. I would really want to see the protocol ahead of time. I think I'd even want to get a sense of the study that he has on childhood leukemia that has been approved by the IRB.
How did you get that?
Do I have to tell you it's not approved?
And then I guess I definitely would want to know more about the state, Pennsylvania's newborn screening law. I am not an expert in newborn screening. Most of my genetics is kind of in the adult spectrum, but I know it's a really kind of hot area right now. And I do know that investigators in Minnesota are really quite upset with what has happened of late at limiting the use of the newborn screening. So it's kind of, I mean, I think the message that I was trying to get across is you have to balance enabling research and allowing it to go forward, but still maintaining and building the trust of the public and policymakers, because I increasingly think that that's more and more fragile, and I don't like investigators to see me as a stop sign or a roadblock, but as someone who kind of wants to work with them and gets just as excited about the science as they do, but wants to make certain it's happening in a responsible, thinking about socially responsible way.
Questions or comments people have?
So, hi Jen, I'm Trish Gordon from the IRB.
You guys.
Yeah, I'm one of them. I should also disclose that I'm a clinical geneticist and I'm the point person for newborn screening here.
Okay.
So I'm not trying to throw a whammy at you. I'm just trying to, so in the state of Pennsylvania, just for the record, they will not allow any research on the samples. In response to Minnesota, Texas, and so forth. They used to store them for years and years and years for us, so we could go back and say, well, you know, we now have another child in this family. So now I think they hold them for 4 weeks and then they destroy them. So unfortunately, Kent wouldn't be able to get his blood samples anyway. But I was intrigued by your suggestion for a summary explanation of research and an opt-out type of situation for prospective consenting. What would you think Dr. Verana's obligations would be to the public to, as you said yourself, a lot of times people aren't really quite aware that the newborn screening is happening anyway. So to me, once we take it from a clinical to a research realm, we have a higher bar. That's my opinion. I think that's Kathy's opinion too. What would you think his obligations would be to make the public aware of what was happening?
Of the fact that he was using.
If he was going to do this summary explanation of research opt-out type of scenario?
Public at large? It's an interesting question.
I guess my question is, do you think it's adequate to try to do it on a one-on-one basis? Or do you think it's something, if he's collecting from 20 different hospitals in the region, should there be a public campaign, something?
Sort of like what you would do with emergency medical.
Right, or like what Vanderbilt did, something along that line.
Yeah. I mean, that's actually an interesting approach. You could have, so kind of even stepping back a little bit, I was involved in an emergency medicine critical care research study, and the investigators came to me wanting some insight on how to get public input, knowing that town halls don't necessarily do well. And so what we came up with was a model of doing focus groups, so kind of, I guess what you could say, mini town halls. And we did them not just at Mayo, but also Olmsted Medical Center, which is another hospital health center in the city. And so if there were resources and if there were a desire, I could see doing a number of mini type focus groups over a short period of time to actually get the potential participants or parents of the participants input on what they would like to see. Because I think one of the things that I've come to appreciate is a lot of times we as the experts can stand back and, you know, wave our hands and say, this is what should be done, this is what should be done. But when you actually go and talk to the people who are going to be impacted, they are maybe a little bit more comfortable with things than what we appreciate.
I absolutely appreciate the question of whether the resources are available and so forth, but lack of resources doesn't necessarily negate the ethical issue here, and we've already identified that knowledge and understanding of newborn screening is probably limited.
Right.
Now we're going to take it to a research step. You've also got all different types of cultural issues in the region with the Amish and the Mennonite population. We have immigrant populations, things along that line. So my question is there an ethical obligation to take this further than just here's the paper, you want to do it or not?
Again, I'm not, you know, I'm kind of, I'm not sure I know how to answer that question. And what's going through my
That's
mind.
fair. I appreciate those kinds of
Well,
answers, very much so.
I want to actually go back, and what's kind of basing my statement on that is the state of Minnesota, which some of you may know, has a law that we cannot use patients' medical records for research unless we've obtained authorization. So it's unique to Minnesota. And what's interesting about it is that Minnesotans could be in any other state and their medical records would be used for research without their authorization. They would have no say in it, they would have no knowledge of it. And so in Minnesota, we have the what we call the Minnesota authorization, causes researchers a lot of consternation. And I have conversations often with our research subject advocate who we discuss, yes, the research authorization is, something that fosters transparency, empowers autonomy, et cetera, but does it really help, and does it really maybe raise more consternation and issues and concerns than what it's worth? Because a lot of people get upset when they realize it causes a lot of problems amongst our research participants, and I'm not sure it's actually worth what it's trying to get across.
I mean, I think we've had these kinds of true of HIPAA in general.
Yeah, and I think,
You sign that form and you go through the drive-through at Walgreens and they, announce your birth date and your home address over the loudspeaker. So, you're right. I mean, is it really accomplishing what it set out to accomplish?
and that's why when you started talking about, kind of town halls or public notification, it could actually create more scare, more harm, and really not empower participants.
In the debrief, after the mock thing was done, you said that it's really important to try to communicate that you're really excited about the science and you're not a stop sign and you want to move this forward and you're an ally to the research and so on. In the actual interaction, I'm not sure that I got so much of that message from you to him, and I'm just wondering how you might see yourself communicating that along, obviously along with all the ethical concerns that you're raising. How might you sort of ally yourself?
And I think that's one of the things by getting protocols and studies ahead of time that does allow you to be more familiar with what the researcher's doing and to be more excited about it. I think this morning it was probably challenging for me to do that because I had an audience of people and I kind of felt like I was performing, which is not typical for research ethics consults.
No, that's how we're running here.
I mean, usually you're sitting in someone's office or you're at a coffee shop having coffee or the cafeteria, kind of discussing things. So I think my approach is usually typically more trying to get to know the investigator a little bit and what they do generally, what their research is overall, and maybe trying to tell them a little bit about what I do as well. So this, not only was the case maybe was a little contrived, but I felt like the whole interaction was, but that's okay.
So let's explore for a second this nuance about returning incidental findings. Isn't that just at odds with the entire principle that if we use de-identified samples, the understanding on both sides is that I will not be able to track you down, nor will I be able to return them. How do you feel about that?
Well, I think there's an increasing interest in participants in research, and various survey studies show this, and even some of the qualitative data, that when people are participating in things like biobanks or repositories, that they are actually expecting something in return, especially if it's information that has some real potential clinical information, usefulness for them. Something like a BRCA finding. You're right. Many women choose not to be tested for that when they are in a family with a strong history of that. That said, in many of the hypothetical situations, and that's kind of a problem with a lot of these studies, is they're hypothetical. If people do actually want to get that information back. So, and I think, I'm not sure people, we're saying things are de-identified, but I think there's also this increasing awareness that things are never really de-identified, and that you can go back and link. I mean, like, you could relatively, as the investigator, you may not have the key that links things, but someone will, and it wouldn't take, sure, it's me saying this, it wouldn't take too much effort, but it could be done to re-identify, relink people. What's striking to me about this also is that a lot of times with these, the use of DNA samples in biorepositories is that there is actually a desire to have some phenotypic data from electronic medical record. That's what actually kind of brings some wealth to it. And so in doing that, you are actually, you have identifiers with that.
No, I agree with that. It's just, there's a conflict, an inherent conflict, that when I go to our Institute for Personalized Medicine Biobank to get specimens, I'm not provided with the identities, and I can do my studies, and if I happen to find something, no one says, oh, you have to tell me. Just because example would be, I'm sequencing a big chunk of DNA for some other purpose, and I get a BRCA signal or something like that. I'm not told that I have to go back and say, oh, this sample has that. It's part and parcel of the arrangement. Anonymization goes both ways.
Maybe, that's not what I'm familiar with. I think at the biobank that I'm familiar with, when you are requesting samples, you actually have to have a return of results plan. And it is actually kind of vetted by a small group, a committee.
That's not the case here, is it?
Yes, it is.
It is.
The IPM does have a policy, and they have, in their protocol, they have said there's a committee. Now, the investigator who discovers the incidental finding would have to submit a form along with all the information and to this committee to determine whether or not it should go back. And the consent form actually states the possibility of discovering incidental findings. And if they do, the consent form says, if we do, then we'll contact you to see if you want more information.
So we don't ask if you want to be contacted, we say we will contact you.
So the contact basically states, we found something that may be informative, and asks, do you want further information?
Or it actually says, well, maybe that, or in some instances, it puts the burden on the participant to come back and contact you if they want more information.
Right.
If you want more information, call this number.
Well, so the problem, of course, is that then it requires that the investigator who's sequencing a big chunk of DNA actually look at things outside his or her.
So that would be secondary findings. Incidental findings, in the true sense, are really, they just, you weren't looking for it, it just happens to pop up.
I see.
Yeah. So most biobanks that I'm aware of aren't asking investigators to go out and actively search for things. It's just in the instance if something does happen to pop up, because it can happen. And, it may not be common, but in those instances when it does happen, it can give some investigators pause.
This is more of a law question than an ethics question, but are you aware of any liability issues when an investigator found significant incidental finding, did not return the results, something bad happened to the individual that maybe could have been prevented, and where an investigator or an institution was sued for not disclosing?
Yeah, that's an interesting question. I'm not familiar with any, and I think what's interesting to me is institutions' legal offices and legal counsels haven't really caught up with this idea that, this might be a liability issue. So I'm not familiar with any such cases, but it's an interesting issue that I really think does need to be tackled, and I think not just in the context of research, but even in the context of offering clinical whole exome, whole genome finding, sequencing. This idea that institutions may end up with all these data from their patients that is actual clinical data, and they're not, the question is, what's the obligation of the institution to go re-interrogate that data once a year, every 18 months, and update their findings and report that, anything new to the patients? I'm not sure we have an answer to that legally, and ethically I've heard different folks who are much smarter than I am in this area debate both sides of the issue. So it's an interesting question.
Well, it's particularly interesting because so many of our findings provide risk assessment, not causation. And so your example of a hERG channel mutation that gives you a clear risk of sudden cardiac event, that's one thing, but ApoE4 allele frequency, that increases your chances by twofold or something
Right.
of Alzheimer's disease.
And so I think, a lot, there are a number of recommendations, papers that have been out. The one that people often refer to in the context of research, return of results, is Favitz et al. Came out in 2010, and I can't remember the journal. Susan Wolf et al. Has an updated one specifically for biobanks in the journal Law, Ethics, and Medicine. And a lot of times these types of findings are put into different bins. So there's the bin that really you should offer, then a bin that, well, you could offer, then a bin that don't offer. And I think a lot of people, a lot of experts, not patients or research participants, feel that something like ApoE4 would be in the bottom bin.
Yeah, no.
Yeah, especially in a research context. So it's an interesting question too. A number of biobanks, when they have these policies in terms of return of results, they've set out a working list of the types of findings that they would ask their investigators to offer. Not that they would ask their investigators to look for, but if some of these come up, then we would suggest that.
So that would have happened if I said, in a protocol that I was going to sequence the usual suspects, then I would have had to have had a provision in there for return. That makes total sense.
So one of the things that you mentioned, I think, very nicely was the balancing between moving science forward and sort of maintaining or nurturing the public trust. I don't know if there's any expectation that you would go out to the community at large, but do you have any thoughts about the role or the value of nurturing that public trust outside of a particular research protocol? But just, you know, there are a number of reasons why the public is distrustful. Among them is that someone is making a bunch of profit and they're getting nothing, right? And also that something can be done nefarious, but there may be just other things. And I'm wondering if you have any particular thoughts about the value of approaching the community or ways to approach the community to build that trust.
Yes, I am known to get on my soapbox and talk about the civic scientist. And that's a term that was, Neil Lane is given credit for coining the term. Whether he actually did or not, I'm not sure. Neil Lane is a physicist. He was also the second presidential advisor for President Clinton, among other things. And his definition of a civic scientist is a scientist who is willing to go out beyond the walls of their laboratory, their institution, their ivory tower, and talk to individuals or groups, and not doing things like Alan Lecter may do at the AAAS meeting or the Society for Neuroscience meeting, where there's lots of people and he's on a stage and he's talking about the role of science and the importance of science and research to the public, but actually going to the local Rotary clubs over lunch and talking to them about just the importance of basic research. Not necessarily about his research or her research, but why doing science is important. I also think a lot of people who don't have any relationship to someone who's in science really fully appreciate what it means to do science. And that it doesn't happen in a short period of time. You can't start year one and expect to have a deliverable, concrete deliverable that's going to improve humankind in 5 years. So when I talk about the civic scientist in classes I give, I'm talking about just really being comfortable and talking to your neighbor. I share a story that I talked to my Uncle Bill over Thanksgiving about, he was a very educated man, but slightly misinformed on how human embryonic stem cells were derived. And so rather than getting into the,
Family.
rather than just getting up and leaving, or ignoring it, or getting into a political debate, I said, Uncle Bill, I really don't care what your ideology is about this, but can I at least sit down and show you the biology of how these are derived? And because he's my uncle and I'm his favorite niece, he allowed me to do that, and we sat down and spent about 20 minutes, and I kind of drew out schematics and answered questions.
You could consult for this.
And in the course of that, it was kind of interesting because a couple other people who were around came and kind of were looking over my shoulder and not really asking questions, but clearly trying to get something out of it too. And to this day, I have no idea what Uncle Bill thinks about human embryonic stem cells. I really don't want to know. But at least I know I did a small part in trying to make him a little bit better informed. So, it's really little things, but I think the little things can add up. A class that I teach, or have taught, we have a session that, a new session that I implemented, and it's called, what I ask the students to do is to kind of come up with a type of elevator speech And then when they come into the classroom, they do a role play. And so one student, they decide whether they're maybe in the grocery store line waiting for checkout or on a flight across country or whatever. And then so the student, there's one student who has to kind of play the naive non-scientist and start asking questions. Oh, about, what do you do? Oh, that's really interesting. And then the student who's kind of in the hot spot has to go through the process of trying to explain his or her work to that individual who is supposed to not be a scientist.
Questions or comments? Steve, you're a young researcher doing work in epigenetics. Any thoughts or questions about sort of how this kind of consult would figure into your role?
Sure. I guess part of my own personal research spin on it, I do work with epigenetic factors, so not quite people's genes, and with a lot less, at this point, clinical implications, but sometimes run into the same barriers in terms of we blanket impose similar restrictions on the research for microRNAs, which is what I look at. So just curious, I guess, your take on, because it's a spectrum of DNA, RNA, and then proteins. So just thinking, like, as we collect patient samples, how would you see those three uses being different and how we need to consent people?
It's an interesting question. So uses of the samples for looking at RNA studies, looking at proteomics, doing proteomics. I think, well, I mean, anytime you're doing a study where you're collecting samples, and presumably it's probably you're collecting it and you may have a specific research question at hand, but you're hoping to actually keep the samples for future use. I mean, I think in that context, the consent language is pretty similar. I mean, you want to make it clear. Well, I mean, there's two approaches. You can either ask the participant if they'll allow you to keep the sample for future use, or you can just kind of make that a requirement for participating in the study. But either way, it needs to be really pretty clear and part of the consent process, not just the document, but the conversation. I'm not sure I know enough about technology in terms of using microRNAs, but presumably you need to derive the, isolate the DNA first, and then, okay.
So let's just say in a hypothetical context, you're going to only look at RNA and you're going to throw the samples out afterwards, destroy them, you're not going to look at any DNA. The way you collect samples is collecting them in, with a fluid that preserves RNA. DNA immediately pretty much gets degraded and is difficult to get out of that sample unless you have a specific mix of chemicals in your collection vial. So you're left with only RNA, which could potentially code for proteins or potentially code for nothing. That's what microRNA is, it doesn't code for anything. And it's not really part of the human genome per se. It sort of rides that fence. So it's a tricky thing to explain to patients without a scientific background, kind of like you mentioned. But it's also a very hazy gray area in terms of the ethics of what my responsibility is as a researcher to provide information back to people.
In terms of return of results?
Yeah.
So if you are able to identify a particular protein and microRNA that expresses protein. Is that going to tell you anything clinically about that patient? Isn't it that just like a moment in time, or is it? I don't. That's a really good question.
Yeah, so it's exactly, look, at least that's my personal take on it, that it's a specific moment in time, and it's not a fixed expression of their E that stays with them.
Yeah,
Of course, they're like,
and I
yeah,
think because it is a research context, and we really, it sounds like we don't really know what we're doing, we're learning, that it's probably better not to return to patients, unless there's something that is really clearly a red flag. I mean, that really just hits you, across the face. This is important, and even though it's just a moment in time, it is something that may be recurrent, or something like that.
Well, so it's a moment in time, but if you convert that RNA into a cDNA and sequence it, there'll be enough polymorphisms there you could identify the patient if you had the patient's DNA. So that's one of those what ifs. But we have no clinically actionable polymorphisms in microRNAs that I'm aware of that you would return those results. It's just what is their profile at that moment in that clinical phenotype. So there's nothing really to return. But an example would be some work we're doing in protein biomarkers of alcohol, prior alcohol use and abuse. Now that's a different matter, because we could, in fact, we're starting to think about how we would deploy that. You say you drink this much, your blood suggests maybe you're drinking a little differently. And so that's going to be clinically helpful ultimately, but it's not actionable yet. But still, that moment in time could be diagnostic at some point.
Sounds like you need a research ethics council.
Yeah, I mean,
I'm not taking,
that's actually really an interesting question, because then what if you do find something that is clearly divergent from what the patient says, that is so alarming that you might want to contact the patient's primary care.
well, there you go.
Yeah, that's
There's no-
really,
We're not there yet.
Just throwing it out there, Kent, there's no way you should be doing that without consent.
Absolutely. You
Right,
get
so that,
point.
to me, that, you set it up appropriately on the front end, and that, because to me, there are legal all different types of ramifications there, and so that one's easy.
Yeah. No, you have to have consent to do that.
well, yeah, you have to
Yeah.
have consent to do that, but what to do with the finding,
Right.
it's an interesting question, I think. And I don't know how, are you, it raises a lot of questions. Are you recruiting participants who get their health care here, and you would know their primary care docs, or?
No,
are they-
in this case, it's a very small subset to see if there's anything that could be predictive, and it's through a VA that we have an IRB approval for. But I'm totally blind to everything about those patients, except that they're recovering alcoholics. That's the only thing I know.
Well, thank you very much. We'll give you a best.
Thank you.
Thank you.
Well done.